Good afternoon, everyone. I will give you an overview of latest activity of the Solar Imaging Core Facility or SELIM. SELIM is part of a large research infrastructure, Czech Bioimaging at national level. And through the Czech Bioimaging, we are obtaining finances for running costs and also for investment. And therefore, I will repeat the previous colleagues that it is very important to use acknowledgement in publication, in articles, if you are using our facilities, because this will gives us money and for investment and for running the instruments and to provide the services for you. So thank you for all of you who are already doing it. And at the European level, we are part of this research infrastructure, Eurobioimaging, which are connecting together imaging facilities across Europe. In terms of staff, we are 4 people. So it's me, Petra, who is taking care most of the time wide field microscope and confocal microscopes, and he is the person also who will run the light sheet microscope. Then is Kuba Pospíšil, who is operating as well the wide field confocal microscopes and he's expert for super resolution microscopy. And we have Wojciech, who is also doing the wide field imaging, and he's expert for image analysis and data analysis. In terms of users, we get in last 2 years a little bit less users in total compared to year before COVID. We will see if we'll increase again in this year or next year. In terms of hours, there's still increasing numbers, so people are using more, and we have also more instruments. And in terms of publication, we had a little bit less publication than the years 2019. Here's the slide how you can contact us. We are located in C2 building. Our headquarters is in C2 building, and the part of our microscopes is at E26. And if you want to reach us, you can come directly to our office, or you can either use this email, the selim@cetec-muni.cz, or you can use this email, selim support@cetec-muni.cz. You can also reach us directly from our website. So we have there implemented the SELIM help desk. You can just click on it and you will write your name, email, and what is your requirement. And it will create directly the ticket in our internal database. And so then we can contact you back using this system. We updated slightly our web pages so that they will be standardized for all core facilities of CETEC. So we have there the user access and there are all important information according the user rules, user training, and the access to booking system. So the first part is the user training. So there's just information how user trainings are provided and what do you need for user training. And there is also directly the button you can click and you can write that you would require the training for some of the microscope, and we will contact you back, and then we can find some date, and we will select the instrument most proper for your requirements. In terms of user rules, you can find also the user rules on the web pages. So I would mention just recently we had to implement the quota system for use only with confocal systems, because there was many people using confocal microscopes, and there were people complaining that they are not able to use it during the day. So we implemented that every single user can use maximum 4 hours per day from 9 to 5 p.m. And outside this time window there are no limits for using the microscope. Therefore, if you need some longer experiments, it is better to plan it for afternoon or overnight or during the weekend. So there are on national holidays. So there are no restrictions to use the confocal microscopes on the weekends and national holidays. Yeah, and the quota is valid only for confocal microscopes. No other instruments have quota for user usage. In the term of events, so we are organizing a lot of events, workshops, and courses. And so they are always advertised via the CEITEC websites and the CEITEC PR office. And we have also advertising them on our web pages, so you can find it there. And we are using also Twitter. So our facility has an account on Twitter, and we are posting the important news and events using this social network. So if you would like to hear the new news from our facility, you can subscribe on Twitter, and you will have them from first hand. And so here I would like to draw attention. So at the beginning of November, so we are having our annual light microscopy course. So you can just still subscribe to this 3-day course of light microscopy. In terms of booking system, so as other facilities, so we are using the CEITEC booking system. So you can use it. You can access the booking system directly from our web pages. And so new user, they can first time register, and then they can access the booking system to book the instrument of the facility. To see just in the better way the usage of instruments, so we are using this web page. You can see here the today.ceitec.cz/selim. And there is nice overview of instrument booking during the day, and also it's during the week. So you can look how the single instrument are used, and you can book after it, like via the standard booking system, where the overview is not so nicely done as the previous web page. So here is our price list. So it didn't change since last years. So for internal CEITEC user, there are these prices. If they are registering to Czech Bioimaging database, and then the requirements is to, of course, to do the acknowledgement to Czech Bioimaging. So I will put also the prices are very reasonable, and it was also from the survey, so that no one was complaining that the prices of our microscopes would be too expensive. So new instruments. So in last 2 years, we purchased 2 big new instruments. So first of it is this new Carl Zeiss ELIRA 7 super resolution microscope, using the LATISSIM and single molecular localization microscopy. So the LATISSIM is extremely simple and very powerful technique how to obtain the better than Abbe diffraction limit limited systems. So using this this technology or these methods, you can use the samples. There is no special requirement for sample preparation. You can use the same samples as for wide field or confocal microscopes, and you can obtain these nice images reaching the resolution up to 60 or 80 nanometers on XY axis. So this is really, really amazing. Here's some fibroblasts you can see, and this is from Misha Procházková from Pavel Plevka lab. So the Staphylococcus bacteria. These are 1 micrometer of size, and you can really see the membrane labeled and the DNA labeled. So there's a very nice details. You can use also the single molecule localization microscopy. This is a more tedious technique, but it allows you to reach much higher resolution, closely reaching 20 or 30 nanometers on lateral axis. But it is not as simple as the SIM microscopy. Another big system is the light sheet microscope, and this is a completely different way of microscopy, how you look at the sample. So there is a big attention is on the sample preparation. So it's not like that the sample is on the standard cover slip and you put it on microscope, you are looking at it. there it's you will spend a lot of time with sample preparation and sample mounting into the microscope. But this is a really great systems, and this is we have all objectives and chambers also for clearing solutions. So people using it a lot of large objects, also for organoids and for whole organism. There's mouse embryo, and also you can image the whole plant system, roots, and so on. We also updated our IT infrastructure and system. So all microscopes in C2 buildings, they are now connected with 10 gigabytes data link directly to our data storage and image analysis server. So you can save your images directly on this server, and where you can analyze them also. So the major image analysis softwares, they are running on this server. So we have the Zen Blue with all the modules, including deconvolution, area scan processing, structure illumination, microscopic processing, and so other. We will run the Imaris software soon on this Windows server as well. And we have other open source softwares like Cell Profiler and ImageJ. So that's the big advantage. So you can save the data directly on this server, and you can directly analyze them or post-processed. So we have also implemented these new data management rules of our facility. So where is in briefly, it is also on our web pages, and it is saying just that we are not taking responsibility for data which are saved locally on the microscope computers. That you should, if you are saving the data there, you should remove it directly to your server location, or you can use our this Odeon server, and you can save the images directly on this server, where we offer standardly 1 terabyte of space for 30 days. Yeah, this is not long-term usage, but it is for the time necessary to analyze your data. And after this period, you should move it on your location. For light sheet microscopy, so we do it on individual basis. So we are offering more data storage than it is this 1 terabyte. So here is also some output from the user survey of facility. So yeah, thank you very much for those who just filled these user surveys. And it was mostly positive. So this is the how you are satisfied with the service. Yeah, so there's the majority is there are 1 or 2 very satisfied. There are some they are not satisfied at all, which there was nothing written in the written comments. So it's difficult to judge whether there was a really some problem with our services because then there was nothing written in the written form. So yeah, I don't know how to, whether we can help or something, whether there was some problem. So and from the written comments also, so people were asking for BSL2 samples, if we can if we can image the BSL2 samples. So we can do in limited way. So our laboratory or the microscopy laboratory can handle BSL2 sample, but we have to book all the other systems. So there can be only this user at present at one time. And ELIRA, the super resolution microscope, for the moment is not able to be operating in a BSL2 regime. So we are solving this issue, and it might be possible in future. People would like also have a wider selection of microscope. Mostly they are writing for spinning discs and light sheet microscope. So the light sheet, it is now operational, and the spinning disc we don't have. It is in the plan for future investment. So we will see how it will be in future years. And people would like also to have more space for sample preparation. Mostly, if they would like to have incubators and instruments for live cell preparation. Yeah, so of course, so we have very limited space in the facility. So for the moment, this is not possible to offer you some place where it would be possible to prepare live cells and some more laboratory space for your sample preparation. So there is limited space. So if you need more space, you have to prepare the sample in your laboratory. And people, there were some comments about the image analysis and custom written software. So we are doing the training for the standard image analysis procedure using the Zen software and Imaris. We don't do the custom written software, but we are collaborating with Michal Kozupek from Faculty of Informatics. And if there is some requirement to write or to make some customized software, we could we could connect you directly to him, and he might be able to help you. Yeah, so I mentioned those. This is the acknowledgement. So you will find on our web pages acknowledgement for Czech bioimaging projects, how to write the proper way, the acknowledgement. And also we should think also about co-authorship when the people are more involved in the sample preparation or sample microscopy or image and data analysis. So here is a selection of articles. So we have the users that are publishing in very good articles. So like this one is Angewandte Chemie from the Lukáš Trantírek. We have from Marcela Buchtová in Scientific Reports. Yeah, the paper from Lukáš Čánek laboratory in Journal of Cell Biology. We have also in Nature Communications. This one from Nicola Silva from Faculty of Medicine, and here is from Jan Křivánek, also from Faculty of Medicine. And I would like also to draw your attention for the CELIM, the Best Microscopic Picture Contest. So we organized last year the first edition. So here are the pictures. They were selected as the best one. So the winner was Jakub Sumbal from Faculty of Medicine for this organoid picture. Then was Sara Di Atuluri from Helen Roberts group, and Nela Jandová from Marcela Buchtová group with this mouse embryo. So last year, so we got very limited prizes. So we printed this microscope on 3D printer and some cup and coffee and some posters. And but this year we asked Carl Zeiss, and they will provide not microscope, but they will buy this GoPro Hero 10 camera for winner of the best picture. So just it will be open at the end of the year. So if you have some nice images, don't hesitate and send it to us, and you might win this great camera. And that was the last slide. So thank you for your attention, and yeah, I will take questions. Yeah.
I wanted to ask you, what is possible to do on the light sheet? What are the objectives? What are possibilities to try on that microscope?
Yeah, we have everything. So we have the objective here from 5x up to 20x, but there is zoom. And so this is the water immersion objectives. So you can put everything. So there is, I think there's no limit. There are no limitations. So most of the time, people they have organoids or the roots or plants. They are putting just you put it to agar, or just you stick it on some holder and you image it. So there's a difficult to say that if there is some limitation. The limited is resolution. You don't obtain as great resolution as with confocal, of course, but you can see much deeper and you can image much larger objects. So for instance, lately we imaged also some mouse embryos developing the teeth very deep, which was impossible to see with standard microscope. Yeah, so this it's really great. And we have also for most people are using the clearing solutions now, because of course the big samples, they are not transparent, so we have to clear them. So there are different clearing solutions now, clearing techniques, and we have objectives and chambers for most of them. So the most used one is the cubic. So that's the most used clearings. So we have objective also for this.
Some other questions?
Just technical question, this OTA, it's in the booking system already implemented for user?
Yeah, no, it's not implement. It's not possible to do it in booking system. I was asking with Tom Bartek if we can do it in booking system. So there is no way, or it's difficult. So we didn't then continue on this. So we are controlling it regularly. So we told the users and
Manual control.
yeah, manual control. Yeah.
Can you elaborate with microscope? What is the phototoxicity of the system compared to normal microscope?
Yeah, it's higher. So we are doing now some live cell imaging, and it's not as, it's much more toxic than confocal microscope. So we have to have a really nice staining, and also it depends how many times you want to image the solution, the samples. Yeah, but it's definitely more toxic than phototoxic than confocal system.
What are the dimensions of the sample that can be taken? How deep you can go into sample?
How deep using the SIM? It's difficult to say in general. It also depends how transparent. So we have to project the grid there. So it depends how transparent is the sample. Yeah. So I cannot say like this. It will depend for sample.
But like normal fibroblast cell, you can go.
That's yeah. So I have this the standard. Yeah, so this is the standard the fibroblast. This is the human fibroblast. So there's no problem. They are very flat cells. So of course, so there's the maybe 5 micrometers or something. No problem.